I did want to mention Graeme. Well, I think Graeme will introduce himself, but he is the co-editor of this book. Amazingly prolific researcher, just an inspiration, absolutely, to me. He's published over 250 papers in top journals such as Nature and PNAS. Has many awards to his name and accolades, and he's going to be giving this next talk. Our beloved co-editor, Jonathan. You want to add anything?
Well, thanks. No, it's just slightly surreal. Graeme was my boss for 10 years. The man doesn't age. I want to know how he does that. He still looks younger than me. Yeah, he is superb, and I think Graeme's real art is taking very complicated things and making them seem like things that we should have known all along. So, yeah, without bigging him up, he's very elegant simplicity is what he's extremely good at. And I know that as someone who came back with a lot of comments on his PhD drafts. So I've learned a lot of the guy, and I still do. I think him and my mother still have more power over me than any individuals on the planet. So, no, it's great to hear from him. And I haven't seen him for years, so it'd be great to catch up.
All righty. Let it roll. Graeme Hayes.
For tuning in today. What I'm going to try and do in the next 15 minutes is talk about one particular chapter from the recent book on ocean sunfishes, and in particular the final chapter of the book, where we tried to bring the communities together to identify some of the key remaining questions relating to the biology of this group. And these sort of exercises are really exciting to do because they bring together a group of experts. So we're getting an expert opinion, if you like, on how the field may move forward in future years. So all of the individual contributors to chapters in the book were asked to come together to identify key questions, which we then triaged and put into this chapter. And this sort of exercise of bringing experts together in a field is something that we've been involved with in the past. Most noticeably, 5 or so years ago, we brought together the movement ecology group. So people that have been involved in tracking large marine megafauna, and we set them the task of trying to identify key questions for that group, regardless of the particular species that you work on. So regardless of whether you track a seal or a whale or a turtle or a seabird, what are the key questions in movement ecology that pertain across those diverse taxa? And that was a really exciting exercise to do. Essentially, it provided a roadmap which is hopefully helpful to the community moving forward in that area. We then followed that up to address a key question that emerged from the first exercise, which was how do we make the most of tracking data to produce positive conservation outcomes? That's something a lot of people in that area talk about. They track animals to help their conservation, but actually realizing conservation outcomes is not straightforward. So again, we brought together experts in the field to identify case studies where positive conservation outcomes had resulted, and we importantly tried to identify the steps involved in producing those conservation outcomes, again to try and provide a roadmap to people working in that field. Another thing that can come out of these horizon scanning exercises is not just to sort of review the area and identify key issues moving forward, but actually to help foster collaboration and data sharing amongst the community. And a great example of that is the recent work led by David Sims, where a large group of people that have been involved in tracking pelagic sharks have come together to share their tracking data. So in this study, more than 2,000 individual sharks were tracked. So this is a sort of study that would be impossible for individuals to do by themselves. It's only by working as a community that this sort of work can be done. Okay, so learning the lessons from those studies, what were some of the key questions that emerged for ocean sunfish research? Well, there were questions regarding the taxonomy of the group that is still uncertain. But there were also lots of questions regarding the life history of free living individuals. So for example, growth rates, longevity, and the ever-present question of what are the likely implications of climate change for the group. So pervading through a lot of these questions was that issue of the free living, behaviour and life history of ocean sunfishes. And pervading across lots of those questions is the issue of, quite simply, why are the adults of so many species of ocean sunfish so large? And I think that addressing that question can help really identify a lot of the issues regarding the uncertainties of the life history and the biology of this group. So how can we get a handle on why ocean sunfishes as adults are so large? Well, perhaps one way of doing that is to look at how key aspects of their life history change as they grow. In other words, what are those ontogenetic changes in their life history? So for example, how do things such as their habitat, their diet, and their foraging success change as they grow? And here a constraint on what's known is the fact that most studies on the free living ecology tend to focus on the smaller individuals, simply because they're easier to get your hands on, they live in more coastal environments, they're easier to capture and handle and equip in various ways. So there's a need to learn more about the ecology of the larger individuals so that this change in life history as they grow can be better resolved. But it's an exciting time to be involved in this research because there's now a range of tools to study the free living ecology of ocean sunfishes. So this is an example from Tierney's work of the sorts of results that can be obtained by satellite tracking. So you can track individuals for long periods of time now. So you identify where they move in the ocean. Are they living more in an open ocean environment versus a coastal environment, for example? And also identify their diving behavior. So you can start to build up a picture of how the habitat changes with body size. And this is perhaps a good example of where pooling of tracking data sets may have great promise in the future. Taking the example of that pelagic shark study that I spoke about earlier. There's also other tools that are available for looking at the free living ecology, stable isotopes, for example, to better understand the diet, as well as animal-borne cameras that are attached to animals, again, to identify what they're feeding on. So it's an exciting time for looking at that free living ecology. So what can we say has emerged from those studies that have been conducted to date? Well, in summary, we can say that as ocean sunfishes get larger, two key things change. Their diet changes and appears to become more gelatinous as they get larger, and their habitat changes. They become more oceanic in their habitat. So what are the implications of those key changes? For example, being a feeder of jellyfish, how does that impact your body size? Is there some sort of link going on there? So with John Houghton, one of the editors of the book, we've reviewed all the different marine taxa that feed on jellyfish and other gelatinous plankton. And in fact, there's a diverse group of marine animals that feed on jellyfish. And they include both small and large animals and a range of different taxa, for example, some seabirds, many different types of fish, lots of different types of invertebrate will feed on jellyfish. But importantly, when you review those studies, lots of these animals are only feeding periodically on jellyfish. They don't feed exclusively on that group. So what happens if we distill down the group of feeders of jellyfish, if you like, and look at the ones that I would describe as obligate jellyfish consumers, animals that feed almost exclusively on jellyfish? And then the list becomes quite small. And one of the classic examples of an obligate gelatinous feeder is the leatherback turtle. And then you start to see parallels emerging between the life history of the leatherback turtle and the life history of large ocean sunfishes. The leatherback's the largest turtle. It's oceanic, it dives deeply, and it has a gelatinous diet. So lots of parallels there, of course, to large ocean sunfishes. So why do I stress those parallels? And the reason is that because of those parallels, we can look at the biology of leatherback turtles, where some things are known a lot better than are known with ocean sunfishes, and then see what has been learned from that species and whether we can apply those lessons to ocean sunfishes. For example, there's been a heap of satellite tracking of leatherback turtles, studies around the globe, and this is just one particular example showing their ocean wanderings. And one of the things that's fairly straightforward to calculate from these sort of tracking data is the animal's speed of travel, which is shown in the frequency histogram. And you can see here that when you look at the speed of travel for leatherback turtles in the North Atlantic, you get this bimodal pattern with a very slow mode of travel and then a much faster mode of travel centered on around 30-35 kilometers per day. And importantly, these different modes of travel mean that the animals are doing different things. In particular, when leatherback turtles are traveling very slowly, that means they've stopped because they've encountered a patch of jellyfish, essentially. So slow speed of travel means high foraging success. But you can see from this frequency histogram that doesn't occur very often. It only occurs about 20% of the time for these animals when they're traveling around the North Atlantic. In other words, for most of the time, leatherback turtles are in transit trying to find patches of jellyfish. So they're fasting for most of the time. So what seems to emerge from this is that if you have a life of feeding on jellyfish, you have a boom or bust sort of lifestyle. You occasionally encounter rich patches of jellyfish, but for long periods you will be fasting. So we can probably say the same thing for large ocean sunfishes. If they're feeding almost exclusively on jellyfish, they will have long periods when they're fasting. So is there a link then between those attributes of feeding on jellyfish, fasting, and large body size? Well, there is, because we know that fasting endurance increases in large animals. And we know that from studies which have looked at the metabolic rate of different sized animals. And this occurs regardless of taxa. For example, a shrew needs to feed almost continuously, whereas an elephant can go without food for longer periods. And in fish, that fasting endurance can be calculated by known measurements of how the metabolic rate decreases in larger individuals. Although those measurements haven't been done on sunfish, they've been done on other groups of fish. And those studies have produced different scaling exponents for how metabolic rate decreases with body size. And those different scaling exponents are illustrated here by the solid black lines, with the values of B being different scaling exponents. And when we're looking at the solid black lines, we're reading off the scale on the left-hand side, so that relative mass-specific metabolic rate decreases in larger animals. And because the metabolic rate decreases, the fast endurance increases, and that's shown by the dashed lines and reading off the scale on the right-hand side. And so although there's some uncertainty depending on which scaling exponent you use, the overall result is the same. Larger individuals can fast for longer periods than smaller individuals. So if we compare, for example, a 25 kilo ocean sunfish with a 1500 kilo ocean sunfish, the larger individual would have somewhere between 3 and 6 times the fasting endurance of the smaller individual. So is the large size of many adult ocean sunfishes linked to the fact that they're feeding on jellyfish, and as a consequence of that, they've got long periods where they have to fast without food? So that's an interesting hypothesis. And of course, to test that further is going to need more information on how those key aspects of the life history change as individuals get larger. So things like their habitat, their diet, their foraging success. Measurements of metabolic rate, which are obviously hard to make, but can be made over a certain size range of animals, at least, to identify what that scaling exponent is. And finally, I hope I've identified the importance of comparing the biology of ocean sunfishes with the biology of other taxa. We can learn a lot by those comparisons with other taxa. So not just things like leatherback turtles that feed on jellyfish, but also other taxa that don't feed on jellyfish. What are the important differences in the biology of jellyfish versus non-jellyfish feeders? Okay, so I hope that's helped identify some of the issues we talked about in this chapter of the book. And finally, I'd just like to thank you all for your interest, and Tierney will be online to take care of any questions you might have. Thanks again.
Well, thank you so much, Graeme, and we have you with us.
Hi, guys.
That's terrific. Yes, and Jonathan too, I think.
1 sec.
Yep. I think he'll be coming. He'll be coming shortly. Nice to have both of you here. I can hear Jonathan. And so Graeme, I got a message. Maybe you were you able to go out into the sea? Lockdown's up right now?
Yeah. Now lockdown is well, the latest lockdown has been lifted this end. So yeah, first thing I did was go for a good snorkel and get in the ocean. So
Yeah.
I think the official in the last lockdown, the official ruling by the government was that recreation was banned. So that was a bit dramatic terminology, but now recreation is allowed. So we can go for a swim in the ocean, which is nice.
Yeah, I think banning recreation is not going to get you reelected. So.
Can you see me now?
Yes,
I can, John.
Graeme, that was very surreal. That took me back to your office. And I've referred to you as being elegantly simple. I think you had to be. So many of those conversations we had over the years, but very nice. Yeah, I don't know how did you not get older? It's weird.
I thought your introduction, John, was a really nice way of saying Graeme's actually a little bit stupid, so he has to simplify things so that he can understand them.
Never. So so you managed to catch some of the day anyway, because I know the time was awful in a way.
No, I did. Yeah, the last couple of hours or so.
Oh, good.
Superb. So yeah, I mean it's amazing, really, when you think of everything that came together there. Like when when Tierney put out the invite to everyone to join us, that it's the ultimate lucky dip of a species. You just don't know what's going to come back, but what came back was awesome. And I don't use that word very often because I'm a bit deadpan, but it was incredible. And I think from editing in it, I learned more than there's things that I hadn't even thought of, aspects of the ecology that I hadn't even thought of. And it's lovely being in that position where you now know how little you know in a way, and then you can really start to answer some important questions. But I used to work on sunfish because they were cool and they were there, but now their relevance is just so much more clear than it was.
They say the mark of a true expert is someone who knows the boundaries of their knowledge.
Yeah, they face me very clearly every day, but I think for this species, absolutely.
Yeah. And I guess one of the things that's come out of several of the talks is that the importance of collaboration, moving forward. You've both made that point really well, and I guess it's a good opportunity to actually move forward on some of those things. So in my talk, I sort of alluded to some of the things that have been done in other fields. So David Sims and Nuno Quirez have sort of notably got all the pelagic shark tracking people together, and that was 150 plus plus people and thousands of tracks. And the picture you get when you look at thousands of tracks in the global ocean is very different to the picture you get when you're looking at 5 or 10 tracks that you might have collected yourself. And those sort of initiatives are now going forward apace, in other areas of marine science. So some of the work I'm involved with Anna Sequeria in Perth in Western Australia, she's now assembled around about 10,000 tracks, satellite tracks of different marine megafauna, so sea turtles and fish and marine mammals and what have you. So the papers that will emerge from that, of course, when you're looking at 10,000 tracks, it's a another level up from the last Nature paper, by David and led by David and his group. So it's something that we should think about as a community. How do we actually make that happen? Everyone that's involved in tracking sunfish, how do we actually get all of the datasets in one repository and get one person, looking at all those with a global view rather than individual studies looking at particular focal sites? And I'm not sure what the answer to that is, but maybe you've got some ideas of how we actually move forward with that.
Well, I think that was that was the point with I asked Laura to put all the tracks that we know of into the movement chapter. So but that's just one snapshot, and more tagging going on now. So it is an interesting place we're in right now. Mayana also brought this up with trying to create a global biobank for genetic samples, so that we can keep track of, the only way we're going to make any headway on understanding the genetic diversity and species diversity is for everyone to pool their resources of genetic samples, and so and follow similar protocols, etc. So I think it just takes someone to take the lead on that and say, I'll be the portal, and then the protocols to follow, so that no one feels like their work is being slighted, and that there's a methodology for citation and recognition of contribution.
Yeah, sure. So I wonder what that when you say get somebody to take the lead, it's the mechanism of doing that, I guess. And with Anna got a postdoctoral fellowship from the Pew Foundation, I think it was, to drive this forward. So it was essentially sort of, she was in a funded position to do all of that. She wasn't just sort of doing it out of the sort of goodness of her heart. So maybe that's something to think about. And the way that we've worked in those projects in the past is that data contributors are part of the science moving forward. So everyone benefits from it, and so there's no exclusion. So the shark Nature paper, I think, had 150 authors on it, and when we've done we did one with other taxa as well in PNAS, and that must have had 50 authors. So it's something that's really happened, I guess, in the last 4 or 5 years, that these big community projects have come to the fore. And you think those, I think, crikey, that's a lot of authors when if you're the lead author and you're trying to put all the addresses together and things like that. It's not it's a big task. But you look in other areas of science, so you look at stuff that comes out of work on particle accelerators and things like that in the physics world, where they're things like from CERN and what have you. And some of those papers in Nature won't just have 100 authors; they'll have 1,000 plus authors on them, because those are projects that are costing not just a few $100,000, not just a few millions of dollars, but a few $100 million or billions of dollars to actually put together. So those things can be done. It just needs a sort of a sea change in people's view, that the people that they might have previously viewed as competitors in that space, for funding and for writing big papers, suddenly you've got to turn that completely round on its head, a bit like John alluded to. You've got to actually view who you may have thought as competitors are actually your allies and your collaborators, and it's working with them. that moves things forward. And that certainly, I've seen that sea change happen in the last 5 years with lots of the marine megafauna tracking people. So I think it's something we just got to get on board with Sunfish and maybe put our heads together. How do we get a funded position for a postdoc to lead that, and then get that portal set up? And it's a bit like that Kevin Costner film. What does he say in Field of Dreams? If you build it, they will come.
They will come.
As soon as you start this thing and you start showing the outputs that are coming from it, before you know it, people are coming out of the woodwork with data sets that you never knew existed.
Yeah.
And so we've seen that with Anna's work, that once you've had, a Nature paper and a PNAS paper, and you say, right, this is going to be the next stage of the work, is a much higher level than those. She has been inundated with people around the world that we never knew had been out there tracking. And John will know, like in the sea turtle community, there's probably 95% of the people that are tracking sea turtles are people that we're not really aware of what they're doing, because lots of those data sets never see the light of the day. So there's probably a lot more data out there than you think just by looking at published papers. We just got to tap into that potential.
I think just chipping on the back of that, Graeme, because that is it's the really positive side effect of how our knowledge is increased. It's not that we're progressing academically; our capacity to write proposals aimed at, because no national funding agency is going to fund anything on that scale. It's just not at the top of anyone's list. But I think, as you said, like Pew and these kind of large philanthropic things are much more likely. But our capacity to write grant proposals now is so much better, because Graeme or me, we've had about 22,000 pass across our desk, and you can't do that. We know very little about scatter plot approach, and we need to know, and we need to know, and we need to know. It's weird. You need to put forward a pressing question, but you also need to substantiate that you've got the tools and the knowledge, and you're not starting from scratch. And I think we could put a really quality proposal together now in a way that we couldn't have done 10 years ago. I don't know if you agree with that, Graeme, or...
No, absolutely. I think that's definitely the way to go. And maybe a starting point as well is just if anyone's got a really good postdoc out there that wants to start off not just sort of digitizing published tracks and showing them on a map, but actually starting to get the raw data from everyone and starting on that road to using the original empirical data sets to sort of add to our understanding. And then you've got the ball rolling, because my experience with these things is that once you've had some success as a community working together, that just breeds more success and more success and more success. So it's getting that initial win when everyone sees the benefit of collaborating, and that initial win might even happen before there's big funding. It might be something we can start now, essentially.
Well, I would love to continue this discussion and really, put pencil to paper and start, itemizing it out. It'd be great.